Okay, so that gets us to this next slide, which is what is the relevance of studying brain pathways and track maps. The significance is, first of all, that axons are only conducting action potentials in one direction. So when we're looking at maps, it's like a one-way system through the brain. And so we can classify our pathways through the brain and through the central nervous system as sensory or motor. So, for example, if we get the pointer, from the spinal cord we have sensory information which goes up to the brain. And in this case, as we'll see later, it's to the thalamus. And so that's a sensory pathway. We will then have a motor pathway. So we'll have from the brain, from motor cortex, we'll have an output which goes down to the spinal cord. Those tracts are the equivalent of nerves. So in the central nervous system, a tract is basically equivalent to a nerve. So it carries information from one region to another. And we give these tracts often, not always, but often names on where they go to. So this one, from the spinal cord to the thalamus, is the spinothalamic. From to corticospinal, from the cortex to the spinal cord. Sometimes they'll be named on where they're going from and to in the brain, so we'll have thalamocortical, from the thalamus to the cortex. Sometimes they'll get more complicated names, like one of the ones we'll talk about next week is the dorsal column medial lemniscus, but we'll talk about that when we get to it. And understanding these pathways, these connections, has been the key until recently in understanding how the brain functions and malfunctions. A couple of examples of some of these maps. On the left here we have an anatomical pathway for motor control. So the pathway is going to start up in the cortex where we've got the blob. It's going to go down through the cortex, through levels of the spinal cord, and then terminate with our less than signs and various places. This sort of diagram is one we'll look at next week, and it's quite difficult because, as I'll emphasise again next week, what we're trying to do is follow something that starts up here in the brainstem and goes down through it. And we want to show where it is in different levels of the brainstem. So it brings together the pure pathway and the region that it's going to, and carries information like here we've got a decussation where it's crossing the midline. Alternatively, when we're looking at motor control, we might look at a very blocky physiological type diagram. So this is the pathway through the basal ganglia for controlling movement before the one we saw on the left. And so it's basically saying the cortex is excitatory of the caudate nucleus, as is the substantia nigra. That excitation leads to an inhibition of the globus pallidus. Now the globus pallidus is normally inhibiting the thalamus, so you can see, hopefully, and Steve will explain this in more detail, that if these excite the caudate and the putamen will inhibit the globus pallidus, so we'll stop that inhibitory pathway, and so this excitation will be allowed to go ahead. So it's building together inhibition on inhibition to get excitation. Another way of looking at a pathway is one like this. This is one from the Sylvius, which is actually a animation, but it doesn't work on the download. So we get a stimulus in the periphery that sends a signal up to the brainstem, no, to the spinal cord, which goes up to the brainstem. We then have a synapse, zoop, and then we have the next cell crossing the midline and then going up to the thalamus, another zoop, and then for the thalamus up to the cortex. Again, this is trying to show you the three-dimensional structure of these pathways, and we'll wrestle with that a little bit next week. So, oh look, hey, it's working. Thank goodness. There, okay. So that's showing an animation of that pathway. So how do we study these connections? There's various ways that people have in the past and continue to study connections. One is we can look at the anatomy and histology of an individual. And so this is a section of a spinal cord. This is fibres. So these are tracts running to and from the brain. And this region in here is not staining. And this was somebody who had damage to their pyramidal tract, which is the major outflow, motor outflow from the cortex, and post-mortally had a look at their cervical spinal cord and could see that the fibres in this region were dead, which means that that output from the cortex wasn't getting down to the spinal cord because it had been damaged at some point. And looking at a number of those examples, we would then be able to say that that had been damaged higher up, as we'll talk about, which is actually I've jumped ahead to that's the tract death. So that's showing that that connection has died. We could look at more complex histology, and so for example, this is a section of the brainstem, and as with the spinal cord, it's divided into regions which are mainly fibre tracts and some regions which are cellular. And we can look at it in a couple of ways. This is a fibre stain, and where we've got tracts, we'll have dark regions. So this one here is a pyramidal tract, which is those descending cortical fibres. There's tracts here. We've got regions which are mixed tract and cellular, but on a section like this, which is the main ones we'll be working with, the myelin fibres are stained and the cell bodies of nuclei are unstained. This one here is a diagram of a cell stain, and when we have a cell stain, regions of cells which have similar functions are generally referred to as a nucleus. And so this region here, which is a lightly staining region because it's cell bodies, is the olivary nucleus. It's part of a complex, it's the inferior part, but there are the olivary nuclei. Similarly, although we can't really tell, there's a region here, which is this group of cells here, and that's part of the hypoglossal nucleus, so 12th nerve part of the hypoglossal nucleus. And so with a cellular stain, we can identify the regions where you've got a lot of cells doing the same function. With a fibre stain, we can see the tracts. And as I say, most of the ones that we'll be working with are looking at tracts. So we can see the tracts in this section, and as we'll look at next week, if we look at subsequent sections, we can see where the tracts go. Another famous histology was Brodmann, and Brodmann sliced the brain up into thin slices and looked at the histology of the cortex in different regions, and can sort of see that there are layers of cells, and he found that in different regions the arrangement of the cells was different between a typical motor region and a sensory region. And then what he did was divide the brain up into 40 odd regions, which are known as Brodmann's areas. We don't expect you to learn Brodmann's areas. You'll come across them, and some of them are useful and some of them we'll refer to. So for example, 17, 18 and 19 equate to the visual areas. And Steve in his lectures will talk about the different functions of 17, 18 and 19. Similarly, 1, 2 and 3 are the primary sensory area, 4 is the primary motor area, and there are others which relate to specific functions. So the structural difference that Brodmann saw does relate to a functional difference, and we can identify that now. As I say, some of them are useful, but not most of them. Again, as I said, we can do post-mortem analysis. We can do dissections. So this is a dissection of connections in the brain, and so we can see where fibre tracts are going. Another area is when brains are damaged. We can, again, post-mortally examine them and sort out what's going on. And so, for example, this is a diagram of the sensory speech area, and basically the initial identification of the sensory speech area by Wernicke was he had a number of patients who had brain damage and had sensory speech problems. They couldn't understand speech, and he post-mortally analysed their brains and found that different people had different regions which had been damaged, but basically all of them overlapped with this area here, and so this region is identified as Wernicke's sensory speech cortex. And so it's from looking at a number of investigations into the post-mortem analysis. Oh, going the wrong way. Boom, 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 boom. And we talked about tract death. Neurophysiological recordings provide some. EEGs give you a general idea of brain function, so obviously in something like an epileptic fit, the activity of the EEG will be different. But another important one during surgery is when people are doing neurosurgery, if a neurosurgeon is, for example, wanting to remove a tumour or something like that, they will record in regions nearby to make sure that they are not cutting out important functions. So, for example, in this region here, they would have identified motor outputs, various sensory ones here. So people can put in electrodes and either stimulate them or record from them and see what's happening, because people during neurosurgery are awake. And so this sort of mapping during surgery has helped to confirm some of the areas that we've identified, so that around this region here, extending to there and there, would be electrophysiological recordings for Wernicke's area. Similarly around here, the motor speech area. As techniques have improved, we've now got additional components. This is a brain dissection showing fibre tracks connecting different parts of the cerebral cortex. We can now do scans which identify the tracts in even more detail. So we can see this tract here running down through there, or this one along here. So the tracts are a lot more identifiable. Obviously neurosurgery, there's some famous cases where neurosurgery either had unexpected consequences or bad consequences, and the outcome of that has again allowed us to understand brain function. Memory, our understanding of memory a lot of it is based on a case where an individual had part of their limbic lobe removed for other reasons, and after that they lost all of their short-term to long-term memory. Similarly, the corpus callosum connecting to the two sides of the brain was cut in a surgery to control epilepsy and led to a split brain, where we actually found some of the differences in general function between left and right brain. With increasing techniques, we now get functional brain imaging with these scans, and we can scan brains while people are actually doing things like thinking about people or animals or tools, and see different regions of the brain which respond. Here we've got, if you're just reading the word table, the visual cortex is active, whereas if you're listening to it, the auditory cortex, speaking it, obviously motor together with the control, and if you're thinking about associations, then more of the brain gets thrown into activity. So these functional brain imaging is allowing us to get an even better idea of what's happening inside the brain itself. So we've got techniques with humans. A lot of studies that we've looked at relate to animal studies, largely because we have more freedom to experiment on animals than we do on humans. So we can remove part of a brain and see what happens. And so, for example, this is a brain map of the visual cortex showing regions which are innervated by a particular eye. We can cut tracts, so rather than waiting for some random damage, tracts can be cut. We can also do tracing studies. So, for example, these are ganglion cells. And so if you take the thalamus, which is connected to the two eyes, you can inject a material into the thalamus and it gets carried by the cells along the axon to where the cell body is and goes into the cells in the retina. That can tell you a couple of things. One, it can tell you the connections because it travels along the axons back to the cell bodies, and so you can see where in the eye those cells are. Secondly, you can find out something about those cells. So here we've got a large retinal ganglion cell with a very extensive dendrites and a smaller cell with a less extensive dendrite. And we can say that both of those types of cells are going to the thalamus. So we can do active studies of animals if we want to. There's also disease models and genetic models. In Lisa will be talking to you about some of the things that we can find out with genetic knockoffs. The advantage of the animals is we can do things to them, which is also a disadvantage. The other disadvantage is sometimes the animal models aren't reflective of humans, and so for many years a descending pathway called the rubrospinal tract was part of most neuroanatomy courses because it had been identified in the cat, but is eventually dropping out of courses because we're finding that it's not present in humans. One that I didn't mention here, we can do neurophysiological recordings, again some more extensive, but you can also inject dye into a cell and then have a look at it afterwards. So after the experiment, the animals are usually sacrificed, as they say. But we will focus on human connections for the moment. Okay, some definitions that we should talk about. Here we've got a section of the brain, it's a coronal section, and it's been stained, so these are cells and these are fibres. So this is grey matter and this is white matter. And you can see in a section of the brain, you know about the cortex around the outside. There are also central structures that we'll look at. On the surface, the brain is folded to increase the surface area. The grooves between the folds are called the sulcus or sulci, and they're the crevices. When they're really big, they're called a fissure, and the bumps are gyri or gyrus. So here on this one, these are the gyri and these are the sulci in between them. We can see them quite clearly on a lateral brain. Again, the difference between grey matter and white matter is grey matter contains the neurons on a unstained section, sort of looks greyish, whereas the white matter contains the myelinated axons. We'll still have cells in there because of the oligodendrocytes, but it's primarily considered fibres. So tracts are in here, so this is a fibre tract coming from brain to brain, so that's the corpus callosum. This is a fibre tract running from the brain downwards. Talking about directions in the head and the brain is difficult. In terms of the spinal cord, we've got dorsal and ventral, and we've got caudal and rostral for the head end. Problem comes that during development the brain folds over, so you're nice and straight like that and then you bend over, and so theoretically the top of the brain is the dorsal surface and the bottom is the ventral surface. We tend to keep dorsal and ventral for the spinal cord, and in terms of the brain talk about anterior and posterior and superior and inferior. So that's the anterior end and this is the posterior, this is superior and inferior. And yes, we could call this rostral, but I think over the course of the next few weeks we'll be talking more about anterior and posterior. So when we look at sections of the brain, they'll be more anterior or more posterior because of this complexity of this bending. Okay, as a preparation for Friday, basic anatomy of the cortex. It's divided into 4 main lobes related to the bones that they're close to. So the frontal lobe, the parietal lobe, the occipital lobe, the temporal lobe and the occipital lobe. They are generally fairly easy to distinguish. They have fairly definable sets of functions. So we could draw a line here and say motor is anterior and sensory is posterior. And then we have some complexities in the temporal lobe, but we'll talk about that as we go through. So that's a lateral view, and on a medial view we can again see the same lobes. Okay, I think that's probably a good place to finish, and gives me plenty to go on Friday. So we